In this topic, we're going to discuss lipids. So by the end of this topic, you'll know what is a triglyceride, what is the difference between saturated and unsaturated fats, what are the uses of lipids, what are different types of lipids, and what is a phospholipid. Lipids are a diverse group of compounds that are insoluble in water, but soluble in organic solvents such as ethanol. The most common types of lipids are triglycerides, sometimes known as true fats or neutral fats, but you do have other important lipids which include waxes, steroids, and cholesterol. Like carbohydrates, lipids contain carbon, hydrogen, and oxygen, but they've got a higher proportion of hydrogen and a lower proportion of oxygen. Now, how do triglycerides get their name? Well, they get their name from having one glycerol and 3 fatty acid chains attached to it. They are non-polar and they're insoluble in water, but they are soluble in ether, chloroform, and ethanol. So let's have a look at what happens when you join a glycerol plus 3 fatty acid chains. When they come together, water is removed. So we say a condensation reaction has occurred. The bond between the glycerol and the fatty acid chains is called an ester linkage. So the lipid is an ester. If you were to break up the triglyceride using water, which is called hydrolysis, you would get glycerol and 3 fatty acid chains. Remember that lysis is to break up. Here's a simple diagram of a triglyceride. You've got glycerol plus 3 fatty acid chains attached to it. So let's have a look at how a triglyceride is formed in detail. You have glycerol molecule on the left, coming together with 3 fatty acid chains, and a molecule of water is removed from each. So how many molecules of water are removed? You've got 3 molecules of water removed. This is going to form triglyceride. Notice that the bond is between the carbon, and you've got an oxygen, so they're each sharing an oxygen. This is called an ester linkage. So you have water removed during a condensation reaction, and the bond is called an ester linkage. There are 3 ways that you can draw or write a fatty acid chain. This way shows you the complicated way where you've written out all the carbons. So you've got a carbon double bond O, OH. This group is polar. Then the rest of the group, which we call R, which is highlighted in blue, can vary. So it can be C7H15. So generally you write as RCOOH, where R represents the variable group. And if you were to draw it, you would draw it like this. C double bond O, OH, with the R group attached. And that R group is variable. Now you've got 2 molecules here, called saturated and unsaturated fatty acids. What do you notice the difference between the 2? Well, you can see that the top diagram, the top molecules only got single bonds between the carbons, so we say that it is saturated. The bottom molecule's got a double bond, so we say it's unsaturated. If there's more than one double bond, we say it's polyunsaturated. So a good way to remember saturated is saturated equals single, SS. Here's another way to draw fatty acids. If you draw them simply as zigzag, so the carbons are at the points of the zigzag, you'll also be able to see that there's a double bond quite easily. Now let's have a look at the difference between saturated and unsaturated fatty acids and lipids. Here you can see an example of unsaturated, which is C double bond C. If you have more than one C double bond C, it's called polyunsaturated. And these have got a lower melting point, and they're usually found in plant oils. They are also better for us. Here's a nice example of a saturated fat. So remember, single bond C represents saturated, and it's usually found in animal fats. We're now going to move on to the different groups that you find in a triglyceride. the glycerol. So most lipids are esters of glycerol. They're called glycerides. So here you can see glycerol. Glycerol's got 3 carbons with an OH group attached to each. Now, what are some of the uses of, or the importance of triglycerides? Lipids perform a variety of functions. For example, here you can see they are a rich energy source. They provide twice as much energy as carbohydrates when they are oxidized. Thus, they are good sources of energy. They are insoluble in water, so they can't be leached from cells. And some lipids are a concentrated source of chemical energy and are stored in the body. So, if you eat excess lipids, what's going to happen to you? You're going to become obese. Here you can see lipid as an insulator, and it provides buoyancy. Lipids are also good insulators and provide buoyancy because fat is less dense than water. The insulators because when you have fat underneath the skin, it's a poor conductor of heat, so it's a good way to keep the heat within the body. Now, they're also a good metabolic source of water. What does this mean? Well, when fat is oxidized in respiration, it gets converted to carbon dioxide and water. The water can be used by the animal. For example, this little desert kangaroo rat never drinks water, and it only survives on metabolic water from its fat intake. So how do we test for lipids? This is called the emulsion test. If you add ethanol to an unknown sample and shake it, then you pour in water, you're going to get a cloudy white emulsion. The cloudy color is due to any lipid in the sample being finely dispersed in the water to form an emulsion. Light passing through this emulsion is refracted as it passes from oil droplets to water droplets, making it look cloudy. So we say a cloudy white emulsion forms, and you don't say a cloudy white precipitate forms. They are quite different. Now, what are different types of lipids that we get? You've got fats, oils, and waxes. Now, waxes are similar to triglycerides, except glycerol is replaced by a longer chain alcohol, making them more complex structures. They're also more solid than fats, and they're used to produce a waterproof cuticle on insects and leaves. You've got complex lipids, steroids. Okay, phospholipids. Phospholipids have the same structure as glycerides, except one fatty acid has been replaced by a phosphate. The phosphate group has been derived from phosphoric acid, so that it forms a phosphodiester link. So have a look at this diagram. You can see that the phosphate head is polar, so it's at the top. It's joined to glycerol by a phosphodiester link, and then the glycerol is joined to 2 fatty acid chains by ester linkages. So if you were to draw it, you draw a circle with 2 zigzag tails. And here's the structure of a phospholipid in more detail, which you don't need to know. You can identify the phosphate group at the top there, and then you've got the glycerol in the middle and the fatty acid chains on the right. Notice there are only 2 fatty acid chains. The one fatty acid chain has been replaced by a phosphate group. And here's another example. So phosphate molecules have got a hydrophilic head and a hydrophobic tail. So we say that the phosphate head is hydrophilic, water-loving, and the backbone of the carbon bonds in the fatty acid chain are hydrophobic. So why is this important? Well, if you look here, the phospholipids form a membrane. So they're very important in membranes. You've got the phospholipids with their tails pointing towards one another and the phosphate heads pointing outwards. This forms a double membrane, and this is very important in cell structure. So it forms the membranes around cells. This membrane The heads are polar, so they are hydrophilic, and the tails are hydrophobic, so they're nonpolar. They point towards each other. It also makes cell membranes quite fluid. So let's look at a summary of lipids. You've got triglycerides, which are formed from glycerol and 3 fatty acid tails. What is the bond between these molecules called? It's an ester linkage. The different uses are energy, metabolic water, insulation, and buoyancy. What's the difference between a saturated and unsaturated fat? Well, saturated have only got single bonds, and unsaturated have got double bonds. How do you test for a lipid? You use the emulsion test. So you add ethanol and then water, and it forms a white emulsion. And then what is a phospholipid? A phospholipid has got a phosphate group attached to the glycerol and 2 fatty acid chains. It's very important in membranes. So it's a good idea to copy down this diagram as a summary of lipids for when you study later. And that concludes our lesson. The end.